Okay, everyone. Today's topic is the pathway for antigen presentation. So now this section actually is the real function for MHC. So this is the beginning for immune response. And previously we learned the types for antigen. So according to the antigen presentation, the pathway for antigen presentation, and we divide the antigen with endogenous antigen and exogenous antigen. So according to the different antigen, we have two different pathways for the antigen presentation. So for endogenous antigen, it is the cytosolic pathway, and for the exogenous antigen, it is the endocytic pathway. So let's see. For the endogenous antigen, cytosolic pathway for endogenous antigen. So as we know, for antigen presentation, the complete antigen was degraded into small peptide. So we call it antigen processing. So for endogenous antigen, so the antigen, for example, the tumor proteins or some virus and produce this antigen inside the cell, and we will mark it with one molecule. We call it ubiquitin. So after this protein was marked with ubiquitin, and now the cells know, oh, it is foreign things, so their proteasome, these big molecules will digest this protein into small peptide. So this is for the degraded antigen or the antigen processing for endogenous antigen. After the endogenous antigen was degraded into small peptide in the cytosol, and after that, this peptide will move into ER, the rough endoplasmic reticulum. So here, this structure we call it TAP. So this like a gate. So this TAP molecule like a gate. So the peptide, the endogenous antigen peptide, passes through this gate, then from cytosol to the RER. So this TAP, what is it? That is transporter associated with antigen processing. So for endogenous antigen, the peptide must pass through this TAP molecule, then come from cytosol to RER, to rough endoplasmic reticulum. So let's think, if we have some mutation for TAP, now this peptide cannot move into this RER. So this patient has some problem, so they cannot present the peptide on the cell surface. So this patient, we call it the bio-lymphocyte syndrome. And some virus is very clever, so they can binding this TAP. So now close this door, so the endogenous antigen cannot pass through this TAP, so also cannot into the RER. So our cells cannot present the endogenous antigen for virus. So in some virus, they have this mechanism, and they can escape from immune response. So this is TAP. This TAP, they use the endogenous antigen will pass through this door and from the cytosol to RER. So this is the pathway for cytosolic pathway for endogenous antigen. So let's see. So in cytosol, the protein was marked with ubiquitin, right? And this proteasome will digest this protein into small peptide. And this small peptide will pass through this TAP. Then from cytosol into the endoplasmic reticulum. So I said, if some patient has the mutation for TAP, so this peptide cannot move into this RER, cannot move into the endoplasmic reticulum, so this peptide cannot present on the cell surface. And some virus can close this door, and this peptide cannot move into the endoplasmic reticulum. So also the antigen cannot present on the cell surface. So this is the key point. The proteasome to digest the endogenous antigen, and the endogenous antigen must pass through this TAP. Now let's see this MHC1 molecule. So MHC1 molecule was in the reticulum, in the endoplasmic reticulum, and they have some other proteins. We call it chaperone. So this chaperone is also a complex. They have many proteins. For example, the ERP57, the calreticulin, and the trypsin. So they stabilize the structure for MHC1 molecule. So after this MHC1 molecule binding with the endogenous antigen, then move to the cell surface. So this is the pathway for the endogenous antigen presentation, or we call this pathway is cytosolic pathway. So for endogenous antigen, the cytosolic pathway, so you should know these 3 key points. So the first one, this antigen was degraded by proteasome. The second, this peptide will move from the cytosol to RER by TAP, right? And this peptide assemble with class 1 molecule, not class 2 molecule. So here, class 1 molecule. So this is the key point for the cytosolic pathway. And for exogenous antigen, the pathway is the endocytic pathway. So let's see a little different. We know for the endogenous antigen, they should produce new protein inside of the cell, in the cytosol, right? But exogenous antigen, they exist, originating, okay? Exist outside the cell. So they already exist. They just by the phagocytes and by phagocytosis, endocytosis, and internalize this antigen. So this antigen degraded into small peptide in these phagocytes, and they bind with class 2 molecule. Class 2 molecule. So that's C. So this is the antigen, exogenous antigen. They exist outside and they internalize into their phagocytes. And here is that actually they were the APC, right? Antigen presenting cells. And this antigen in the endosome. So endosome combined with the lysosome. So in lysosome, they are allowed to hydrolysis. So this, for example, the proteases or the nucleases. So this hydrolysis make this antigen degraded, become a small peptide. And after that, this small peptide binding with MHC2 molecule and present on the cell surface. So this is different from endogenous antigen, right? The cytosolic pathway. For cytosolic pathway, the antigen passes through proteasome and degraded into small peptide. But for endogenous antigen, and they move into the lysosome, and the lysosome degraded antigen into small peptide. And the class 2 molecule also exists in the endoplasmic reticulum, but the endogenous antigen cannot binding with the class 2 molecule. Do you know why? Because the class 2 molecule binding with one protein, we call it invariant chain. This invariant chain, and this invariant chain occupy the antigen binding place for class 2 molecule. So later, this invariant chain and degraded, but still remained a small peptide. We call it CLIP. So this CLIP always occupy this antigen presenting place. After this class 2 molecule move from lysosome, endoplasmic reticulum, and after that, this small structure, this small protein, then replaced by peptide. So that's why the class 2 molecule also exists in the reticulum, but the endogenous antigen cannot binding with class 2 molecule. So this is for the exogenous antigen, the endocytic pathway. So for the exogenous antigen, so let's see the key point. So the peptides are generated from the internalized molecule. So this antigen already exists outside the cell, just internalized by the phagocytes, and the invariant chain occupy their place for antigen presenting place. And after that, the peptide assembled with class 2 molecules by displacing this clip. So this clip actually is the structure in the invariant chain, right? So after that, the antigen replace this clip, then binding with class 2 molecule. So now this figure we can see this is both the cytosolic pathway and endocytic pathway, all for the endogenous antigen and for the exogenous antigen. So we can see for endogenous antigen, the antigen was degraded by proteasome, right? And after that, the small peptide move into endoplasmic reticulum, and by TAP, that important molecule. After that, the peptide binding with class 1 molecule, then move to Golgi complex and express on cell surface by class 1 molecule. But the endogenous antigen cannot binding with class 2 molecule. You see the class 2 molecule also exists in the endoplasmic reticulum, right? But the structure invariant chain occupied place for class 2 molecule. So the peptide for endogenous antigen in endoplasmic reticulum cannot binding with class 2 molecule. And after that, this class 2 molecule move from endoplasmic reticulum, then move to Golgi complex, and the invariant chain become clip, still occupy the class 2 molecule, right? And the exogenous antigen exists outside of the cell and internalized into the cell and degraded by lysosome, degraded into small peptide, then binding with class 2 molecule and expressed on cell surface. And for exogenous antigen, they were present by class 2 molecule. And endogenous antigen were expressed on the cell surface with class 1 molecule. So there's 2 pathways for the different antigen. So that means the different antigen come to our body. So they have different pathway to be present on cell surface. Later we know for this endogenous antigen pathway will induce a cell-mediated immune response, and some the exogenous antigen pathway will start for the humoral immune response. So this section we learn the antigen presentation for endogenous antigen and for the exogenous antigen. So this class we learn this pathway, the endogenous antigen pathway and exogenous antigen. So we should remember the 2 pathways and how this different antigen was present by class 1 molecule or class 2 molecule.